the enemy within, the biological betrayal. Imagine, for a moment, that the most heavily protected building in America has been compromised, not by an invading army, not by a missile, not by a hacker sitting behind a keyboard thousands of miles away. Instead, by a single trusted employee. He possesses authentic credentials. He knows every hallway. He understands every surveillance camera. He has memorized every emergency protocol. For years, no one notices him because he doesn't look like an enemy. He looks exactly like everyone else. That is where our investigation begins. This is not merely a story about cancer. It is the story of the greatest infiltration ever recorded, one that unfolds inside nearly every human being, one that has likely happened inside your own body countless times without you ever knowing. Tonight, we open a case file that has puzzled physicians, biologists, and detectives of nature for more than a century. How can millions of highly trained immune cells, patrolling every tissue, monitoring billions of cells every day, miss the one criminal that eventually destroys the entire system? Was the immune system asleep? Was it overwhelmed? Or was it deceived? Like every good mystery, the answer begins long before the crime itself. Your body is not a machine, it is a nation. Nearly 40 trillion cells live within its borders. Each has a profession. Some transport oxygen. Some build bone. Some carry electrical messages across your brain. Others defend the nation against invaders. Every second, millions of cells are born. Millions die. This constant turnover is not chaos, it is order. Scientists call this delicate balance homeostasis. Imagine an enormous city where every citizen knows exactly when to begin working, when to retire, and when to disappear peacefully to make room for the next generation. Every cell follows instructions encoded in DNA. Grow, repair, divide, stop, die. Those commands are enforced by an astonishing security network involving chemical signals, neighboring cells, hormones, and perhaps most importantly, the immune system. If one cell begins behaving strangely, alarms sound. White blood cells investigate, natural killer cells arrive. Specialized T cells inspect molecular identification badges displayed on the cell's surface. Anything suspicious is removed. Most of the time, justice is swift, which makes our mystery even stranger because cancer is not an outside invader, it is one of our own. Imagine receiving a report that a trusted intelligence officer has secretly switched loyalties. No dramatic explosion, no obvious warning, just subtle changes, small lies, tiny deviations from protocol. That is exactly how a healthy cell becomes dangerous. Every day, your DNA suffers damage: ultraviolet sunlight, radiation, industrial chemicals, cigarette smoke, normal metabolism. Even breathing produces reactive oxygen molecules capable of damaging genetic material. Fortunately, cells possess extraordinary repair teams. Thousands of DNA injuries are corrected every single day. When damage cannot be repaired, the cell often destroys itself through a carefully programmed process known as apoptosis. It is biological self-sacrifice. Better to lose one soldier than risk losing the entire nation. But occasionally, a mistake survives, then another, then another. Most mutations accomplish nothing. Some weaken the cell. A few kill it outright, yet every so often, one mutation grants a tiny advantage. Perhaps the cell ignores an order to stop dividing. Perhaps it becomes slightly harder to kill. Perhaps it repairs itself faster than its neighbors. None of these changes creates cancer overnight. Instead, they begin a slow transformation. Like an undercover operative gradually abandoning his original mission. The betrayal unfolds one decision at a time. For decades, scientists believed cancer simply escaped immune detection by accident. The evidence eventually revealed something far more fascinating. Cancer and the immune system are engaged in a long strategic war, a contest of adaptation, an evolutionary arms race. One of the most influential explanations emerged through what researchers call the immunoediting theory. Published and refined through decades of research, including landmark work appearing in Cell, the model transformed our understanding of cancer forever. Instead of asking whether the immune system notices cancer, scientists realize the better question is, what happens after it notices? The answer unfolds in 3 chapters, 3 stages, 3 words, each beginning with the letter E. Together, they describe one of biology's greatest detective stories. The first stage is elimination. Think of this as the security team doing exactly what it was designed to do. Every day, abnormal cells appear somewhere in the body. Most never become tumors, Because they never survive. Natural killer cells patrol continuously. Macrophages consume suspicious cells. Dendritic cells collect molecular evidence. T cells launch precision attacks. To us, this battle is invisible. No pain, no fever, no scar. An entire criminal conspiracy may be erased before the brain ever realizes it existed. You have probably survived countless microscopic cancers already. Not because of luck, but because your immune system quietly solved the case before it reached the headlines. This realization changed medicine profoundly. Cancer is not an exceptionally rare event. Rather, its successful escape is rare, or at least it was supposed to be. Some suspects, however, learn. Imagine an infiltrator who survives the first security sweep. He studies patrol routes, learns guard schedules, discovers which cameras have blind spots. Now the investigation enters its second phase, equilibrium. Perhaps the most mysterious stage of all, the cancer has not won, but neither has the immune system. Instead, they coexist. Months, years, sometimes decades. The immune system continually destroys vulnerable cancer cells. The remaining survivors, those carrying mutations that make them slightly harder to detect, persist. Every immune attack becomes a selection process. The weakest criminals disappear. The cleverest remain. Evolution is no longer happening across millions of years. It is happening inside one human body, inside one hidden cluster of cells. The immune system unintentionally becomes the trainer, teaching cancer how to survive, not deliberately, simply through relentless pressure. Every failed cancer cell dies. Every successful one leaves descendants. The rules of natural selection never stopped operating. They merely changed battlefield. Then comes the final act, the one physicians fear most, escape. The infiltrator no longer hides. He controls the building. Cancer cells begin rewriting the rules. Some reduce the molecular markers that immune cells use for identification. Others release chemicals that suppress nearby immune responses. Still others recruit normal cells to help construct protective barriers around the growing tumor. Imagine security guards suddenly receiving false orders, surveillance cameras transmitting altered footage, emergency alarms disconnected from headquarters. The immune system is still present. Its soldiers are still fighting. But the battlefield itself has changed. Modern researchers often describe tumors as ecosystems rather than simple masses of rogue cells. Blood vessels, connective tissue, immune cells, chemical messengers, even bacteria. Everything inside this microscopic world can influence whether cancer flourishes or fails. The enemy is no longer hiding from the system. It has learned to manipulate the system. That distinction changes everything. For many years, physicians asked a simple question: how do we destroy cancer? Today, another question has become equally important. How do we help the immune system remember who the enemy really is? This shift has fueled one of the greatest revolutions in modern medicine. Rather than attacking tumors alone, researchers now investigate how to remove cancer's disguises, how to reactivate exhausted immune cells, how to expose the infiltrator standing in quietly among trusted citizens. The detective story is no longer about finding the suspect. It is about revealing the mask. As we close the first chapter of our investigation, one unsettling truth remains. Cancer is frightening, not because it comes from somewhere else, but because it comes from us. Every tumor begins as an ordinary cell carrying the same genetic heritage that built your heart, your lungs, your memories, your face. The criminal and the victim share the same origin. Perhaps that is why cancer has remained such a difficult mystery. The immune system evolved to recognize strangers. Cancer learned the far more dangerous art of remaining family. And that leaves us with a question worthy of every great historical conspiracy. If cancer can persuade the body's most sophisticated defense network that it belongs, what precise disguises does it wear? Which secret messages silence the immune soldiers? Which hidden codes allow one rebellious cell to convince an entire biological nation to look the other way? Those answers await inside the next case file. And as every investigator eventually learns, the deeper you follow the evidence, the more astonishing the truth becomes. Part 2. The Disguise Principle. When cancer removes its own identity card, every successful criminal investigation begins with one simple assumption. The suspect has an identity, a fingerprint, a passport, a face, something that proves who they are. Take away every identifier, and even the most advanced security system begins to struggle. Imagine an international smuggling network operating across dozens of countries. Its members are not carrying counterfeit passports. That would be too risky. Instead, they erase the passport numbers themselves. The documents still look authentic. The photographs remain unchanged. The paper passes inspection, but the one piece of information that allows authorities to verify identity is gone. Border officers scan the passport. The database searches for a match. Nothing appears. The traveler smiles. There is no immediate evidence of guilt. There is only uncertainty, and uncertainty is often enough to let someone pass. Inside your body, a remarkably similar deception unfolds every single day, except the border checkpoint is your immune system. The customs officers are killer T cells, and the passport is a tiny molecular structure called Major Histocompatibility Complex Class 1, better known as MHC Class 1, or simply MHC-ON. If the first chapter revealed how cancer infiltrates the nation, this chapter reveals how it erases its own identity, not by changing its face, but by lowering the badge that tells immune police who it really is. For decades, immunologists puzzled over an impossible question. Why would a killer T cell glide directly to dangerous cancer cell without attacking? Was the immune cell defective? Was it exhausted? Or had someone altered the evidence? To answer that question, we must understand the remarkable communication system operating inside every healthy human body. Every nucleated cell displays thousands of tiny molecular billboards on its surface. These billboards are MHC-I molecules. Think of them as transparent identification holders worn by every citizen inside a secure government facility. Inside each holder is a tiny fragment of protein, a sample, a status report, an official declaration saying, "This is what I am making." Every hour, every minute, these molecular displays are updated. If the cell is healthy, the displayed fragments belong to normal human proteins. Everything appears ordinary. The patrol moves on. But if a virus infects the cell, viral proteins appear inside the display. If dangerous mutations alter the machinery, abnormal protein fragments are presented instead. Now the message changes. Something is wrong. Investigate immediately. This elegant surveillance system allows killer T cells to inspect billions of cells without opening them. No doors must be broken down, no walls demolished. The evidence is voluntarily displayed. It is one of evolution's greatest security inventions, unless someone removes the display entirely. Imagine arriving at an airport where every traveler suddenly has a blank passport. No identification number, no issuing country, no travel history, nothing. Security officers cannot instantly arrest everyone, nor can they instantly trust everyone. Their decision-making system begins to collapse. Cancer exploits precisely this weakness. Many tumors gradually reduce or completely eliminate their MHC-I molecules. From the outside, they appear strangely silent. The molecular identification badge has disappeared. To a killer T cell, the result is devastating. It cannot recognize what it cannot inspect. The suspect is standing directly in front of the detective, yet the detective has been denied access to the evidence. No badge, no case, no arrest. This strategy has been observed across numerous forms of human cancer: melanoma, lung cancer, colorectal cancer, breast cancer, and many others. The disguise is astonishingly simple. If your enemy identifies criminals by reading identity cards, hide the card. But every mystery demands another question. Who ordered the disguise? Cells do not make strategic decisions consciously. Genes do. Somewhere inside the cancer genome, someone had issued new instructions. For years, scientists searched for the hidden mastermind. The trail eventually led toward an unexpected suspect, a developmental gene known as SOX17. Under ordinary circumstances, SOX17 performs important functions during embryonic development, like many genes involved in early life. Its activity later becomes tightly controlled. Yet in certain cancers, the switch turns back on. Why? That question remains an active field of investigation. But researchers began noticing something deeply intriguing. When SOX17 became active, another crucial communication pathway became unusually quiet. It was as though one radio operator had ordered every emergency transmitter to stop broadcasting. To understand why this matters, imagine every police station across America communicating through encrypted military radio. The moment a threat appears, headquarters broadcasts an emergency signal. Every patrol immediately receives new instructions: increase surveillance, raise alert status, Inspect identification carefully. Inside the immune system, one of the most important emergency broadcasts comes from a signaling molecule called interferon gamma. Scientists often describe interferon gamma as one of the master coordinators of anti-tumor immunity. When immune cells release this powerful messenger, neighboring cells respond by increasing their defensive capabilities. Among those responses, they manufacture more MHC-I molecules. In other words, interferon gamma tells cells, raise your identification boards, make yourselves easier to inspect. This is exactly what the immune system wants: visibility, transparency, evidence. Cancer, however, prefers darkness. Research emerging from scientists at the Massachusetts Institute of Technology, together with collaborators, uncovered compelling evidence that in some early tumors, activation of SOX17 suppresses the cellular response to interferon gamma. The consequences ripple through the entire security network. If cells ignore the interferon gamma signal, they produce fewer MHC-I molecules. Fewer identification displays appear. Killer T cells receive less information. Recognition becomes dramatically more difficult. The disguise is not achieved by constructing elaborate camouflage. Instead, the radio station transmitting security instructions has been quietly switched off. Imagine every airport receiving a classified message instructing officers to verify passports more carefully. Now imagine someone intercepting the transmission before it reaches the terminals. The officers never become more vigilant, not because they failed, but because the warning never arrived. Cancer's deception begins astonishingly early. Long before a tumor becomes visible on a medical scan, its communication systems may already be sabotaging immune surveillance. That realization transformed how researchers think about the earliest stages of cancer evolution. Perhaps invisibility is not a late adaptation. Perhaps it is among the very first. Yet every perfect crime leaves traces. Nature always builds backup systems. Removing MHC-I creates a new problem for cancer. Natural killer cells, the NK cells introduced in our previous investigation, specialize in detecting precisely this abnormal absence. Unlike killer T cells, which require identification cards to confirm guilt, NK cells become suspicious when those cards disappear altogether. Imagine customs officers who arrest travelers carrying forged passports, while another security team investigates people carrying no passports whatsoever. Cancer suddenly faces a paradox. Display MHC-I and killer T cells may identify it. Hide MHC-I completely, and NK cells may attack instead. The tumor now walks a dangerous tightrope: too much visibility, and it dies. Too little visibility, and it also dies. Many cancers solve this puzzle by choosing the middle ground. They reduce MHC-I just enough to confuse T cells, while avoiding complete disappearance. It is not invisibility. It is controlled ambiguity, the biological equivalent of forging documents that appeared just convincing enough to avoid immediate suspicion. The more investigators uncovered, the clearer the conspiracy became. Cancer is not merely a collection of rapidly dividing cells. It behaves like an adaptive intelligence shaped by evolution, not conscious, not calculating, yet relentlessly selected for survival. Every mutation becomes another experiment. Every immune attack becomes another examination. Every surviving cell carries forward whatever disguise proved successful. Generation after generation, the deception improves, not because anyone planned it, but because biology rewards anything that survives long enough to reproduce. Evolution itself becomes the mastermind. This discovery has transformed one of the central goals of modern cancer research. Scientists no longer ask only how to kill tumor cells. They ask, how do we force them to reveal themselves? How do we restore the missing identification cards? How do we reactivate the interferon gamma communication network? How do we persuade hidden tumors to become visible once again? If those questions can be answered, the immune system may not need to learn an entirely new strategy. It may simply need the evidence returned. The detective was never incapable. The case file had been stolen. Our investigation closes tonight with another unsettling realization. The greatest disguise in biology is not changing appearance. It is removing identity. History is filled with spies who crossed borders under false names, counterfeit passports, altered documents, erased serial numbers. But cancer accomplishes something even more remarkable. It convinces the body's own security system that there is nothing to inspect at all. No passport, no fingerprint, No record, only silence. And in the world of biological espionage, silence can be more dangerous than any lie. In the next chapter, we will uncover an even darker secret. What if cancer does more than hide from the immune system? What if it can recruit the body's own defenders, turning loyal guardians into unwitting accomplices? Because every great conspiracy eventually reaches the same terrifying stage, the moment when the guards begin opening the gates themselves. Part 3, the deadly handshake. How cancer steals the immune system's stand-down code. Every secure nation possesses a secret code. Not a code that launches missiles, a code that prevents them. Imagine two fighter jets racing toward one another in hostile airspace. Radar confirms a target. Weapons lock. The pilots prepare to fire. Then, just seconds before impact, headquarters transmits a classified authentication signal. Stand down. Friendly aircraft. The missiles never launch. Without such a system, armies would destroy themselves. The same principle governs your immune system. Its greatest danger is not that it attacks too little. Its greatest danger is that it attacks too much. An immune system incapable of stopping would become more deadly than any disease. It would destroy healthy organs, healthy tissues, healthy lives. Nature solved this problem by inventing biological brakes, invisible passwords, encrypted stop signals, molecular handshakes that tell immune warriors, this target is not your enemy. For millions of years, these safety systems preserved life. Then one of biology's cleverest criminals discovered how to counterfeit the password, not by breaking the immune system, by convincing it to stop fighting voluntarily. When detectives investigate sophisticated financial fraud, they often discover something surprising. The criminal rarely steals money by forcing open the vault. Instead, he persuades the bank manager to unlock it himself. Cancer operates with the same elegance. Instead of overpowering killer T cells, it persuades them to lower their weapons. To understand how, we must meet one of the most remarkable soldiers in human biology, the cytotoxic T cell, often called the killer T cell. Imagine an elite special operations officer, highly trained, patient, disciplined. Before firing, this officer must satisfy multiple layers of authorization. Is the target truly dangerous? Has identity been confirmed? Will innocent civilians be harmed? Only after every checkpoint has been cleared does the attack begin. This extraordinary restraint explains why healthy immune systems usually avoid attacking their own bodies. Every weapon contains a safety catch. Every trigger requires permission. Cancer learned how to exploit that caution. Long before anyone understood tumors in molecular detail, physicians observed something perplexing. Some patients possessed large numbers of immune cells surrounding their cancers. The soldiers had arrived. The battlefield was identified. Yet the tumors continued growing. It was as though an entire army stood outside the city walls, doing nothing. Why? Were the immune cells exhausted, afraid, confused, or had someone secretly issued new orders? The investigation led scientists toward an unexpected discovery. T cells carry proteins that function as braking systems. One of the most important is called PD-1, programmed cell death protein 1. Think of PD-1 as a highly sophisticated emergency brake installed inside every immune soldier. When activated appropriately, it prevents unnecessary destruction. Without PD-1, immune responses could spiral out of control. The brake exists for a good reason. The mystery was never the brake itself. The mystery was who had learned to press it. Imagine approaching a military checkpoint. The guard raises a rifle. You present official clearance documents. The guard immediately lowers the weapon. Nothing illegal occurred. The guard simply followed protocol. Cancer discovered a molecular version of those clearance papers. Many tumor cells display another protein called PDL1, programmed death ligand 1. When PDL1 binds to PD-1 on a killer T cell, something extraordinary happens. The T cell receives an unmistakable instruction. Stop. Do not attack. Reduce activity. Stand down. The weapon remains loaded. The soldier remains alive. Yet the order to fire disappears. This is not invisibility. It is authorization. Cancer no longer hides from immune attack. It convinces the immune system that attacking would be a mistake. The biological equivalent of presenting perfectly forged diplomatic immunity. One might imagine such deception occurring only occasionally. Reality proved far more astonishing. Researchers discovered that many cancers actively increase PD-L1 production precisely because immune cells are nearby. Picture a spy who notices detectives watching him and immediately produces government credentials. The closer investigators approach, the stronger his disguise becomes. Ironically, immune attack itself can sometimes stimulate inflammatory signals, including interferon gamma, that encourage susceptible tumors to express more PD-L1. The immune system announces its presence. Cancer responds by displaying more stop signs. The harder the soldiers push, the more breaks appear. It is psychological warfare conducted through molecules. Yet PD-1 was only part of the conspiracy. Investigators uncovered another checkpoint, older, and perhaps even more influential during the earliest stages of immune activation. Its name was CTLA-4. To appreciate its role, imagine recruits entering an elite military academy. Before becoming active soldiers, they require authorization, training, evaluation, final approval. CTLA-4 influences these earliest decisions. It functions like a senior officer reviewing whether a mission should proceed at all. When CTLA-4 signals become dominant, T cells remain restrained. Again, this protects healthy tissues under ordinary circumstances. Again, cancer discovered how to exploit the safeguard. The tumor had infiltrated not only the battlefield, but the command structure itself. Few scientific detective stories have changed medicine as dramatically as the investigation into these checkpoints. For decades, cancer therapy focused primarily on destroying tumors directly. Surgery, radiation, chemotherapy. The strategy resembled demolishing a fortress stone by stone. What if there were another option? What if the fortress could be captured by reawakening the army already surrounding it? One scientist refused to abandon that possibility. Dr. James P. Allison, working through years of skepticism and failed assumptions, became convinced that CTLA-4 functioned not as an accelerator, but as a brake. If the brake could be released, perhaps dormant immune cells would attack cancer naturally. It was an audacious hypothesis. Many considered it unrealistic. Some believed it dangerously optimistic. Yet experiments repeatedly pointed in the same direction. Block the brake, restore the attack. Meanwhile, across the Pacific, another investigator followed a different trail. Dr. Tasuku Honjo explored the biology of PD-1. Initially, no one imagined that studying this single receptor would reshape cancer treatment around the world. But gradually, the pieces aligned. PD-1 was not merely another protein. It represented one of the immune system's master control switches. Tumors had learned to manipulate it. If scientists could interrupt that deceptive handshake, killer T cells might recover their function. 2 investigations, 2 separate pathways, 1 revolutionary conclusion. Do not strengthen the weapon. Remove the hand restraining it. The clinical evidence that followed transformed oncology. Patients with advanced cancers that had resisted conventional therapies sometimes experienced something almost unimaginable. Tumors shrank. Some disappeared. Others remained under long-term control. Not every patient responded. Not every cancer proved vulnerable. But for a significant number of individuals, the impossible became possible. Instead of inventing an artificial army, medicine had uncovered a method for releasing the fire fries, the one evolution had already built. This strategy became known as immune checkpoint inhibition. Rather than poisoning cancer directly, these therapies interrupt deceptive molecular conversations. The counterfeit stop signal is blocked. The fraudulent handshake is denied. The T cell receives permission to fight again. In 2018, the extraordinary significance of this discovery was recognized with the Nobel Prize in Physiology or Medicine, awarded jointly to Dr. James P. Allison and Dr. Tasuku Honjo. The honor celebrated more than 2 brilliant careers. It marked a profound shift in how humanity understood cancer itself. For the first time, defeating cancer did not necessarily require inventing stronger weapons. Sometimes it required removing the enemy's stolen password. Yet, every breakthrough revealed new mysteries. Why do some tumors respond dramatically while others resist? Why do certain patients experience lasting remission while others gain only temporary benefit? What additional disguises remain hidden? The investigation continues. Scientists now recognize that PD-1, PD-L1, and CTLA-4 represent only part of a vast communication network. The immune system speaks an extraordinarily complex language. Cancer has spent millions of evolutionary experiments learning its grammar. Every successful mutation refines the deception. Every failed mutation disappears. The conspiracy grows more sophisticated with each generation of surviving cells. There is another lesson hidden within this story. History often celebrates powerful weapons, stronger tanks, faster aircraft, more accurate missiles, yet some of the greatest victories have come from understanding communication rather than force. A forged order, a broken cipher, an intercepted transmission. Wars have turned on messages as often as on machines. The battle against cancer may follow the same rule. The decisive factor is not always the strength of the immune army. Sometimes it is whether the army is hearing the truth. As we close today's case file, the evidence points toward a disturbing conclusion. Cancer does not merely survive by growing rapidly, it survives by becoming an extraordinary student of the immune system. It studies the rules, learns the passwords, steals the emergency codes, and persuades the body's own defenders to obey counterfeit commands. The most dangerous enemy is not the one who destroys your defenses. It is the one who convinces your defenses to destroy themselves through hesitation. But even the cleverest code can eventually be broken. And in our next investigation, we will uncover an even darker layer of the conspiracy. What if cancer does not simply silence immune soldiers? What if it actively recruits entire populations of immune cells, transforming guardians into collaborators until the battlefield itself begins protecting the invader? That is where the mystery grows deeper and where the next encrypted chapter begins. Part 4, the tumor microenvironment, the toxic swamp, where justice slowly disappears. Every great criminal empire eventually learns the same lesson. The safest hiding place is not an isolated cave. It is a city that no longer believes in justice. Imagine Gotham City, not the version where heroes always arrive in time. Imagine the darker years. The streets are poisoned by fear. Police officers have become exhausted. Some have been bribed, some intimidated. Others simply stop believing that the battle can be won. The courthouse still stands. The police stations remain open. Patrol cars continue driving through the streets. From a distance, everything appears normal. But beneath the surface, the city itself has changed. The environment now protects crime rather than preventing it. This is one of cancer's greatest secrets. A tumor is not merely a collection of rebellious cells. It is an entire society, a corrupt ecosystem, a hidden city built inside the human body. And before cancer can truly conquer an organ, it must first transform the neighborhood around it. Tonight, our investigation leaves the individual criminal behind. Instead, we examine the city that allowed the conspiracy to flourish. Earlier, we uncovered how cancer erased its identification badges. Then we discovered how it forged molecular stop signals that persuaded immune soldiers to lower their weapons. Those discoveries explained how cancer escaped detection. But another mystery remained. Even after immune cells entered the tumor, many appeared strangely ineffective. They arrived. They recognized danger, yet they became slow, confused, exhausted. Some even began helping the tumor itself. How could loyal defenders become unwilling collaborators? The answer lies inside a place scientists call the tumor microenvironment, or simply the TME. The name sounds technical. The reality is astonishing. Imagine entering a city expecting to confront a single criminal. Instead, you discover the roads have been redesigned. The air has become toxic. Communication towers have been sabotaged. Hospitals serve poison instead of medicine. Every institution now favors the enemy. That is the tumor microenvironment. For decades, cancer research focused almost entirely on tumor cells. Scientists asked, what mutations does the cancer possess? Which genes are damaged? How quickly does it divide? These questions were essential, but they overlooked something equally important. No city survives because of its buildings alone. It survives because of the people, the roads, the utilities, the economy, the environment. Cancer follows the same rule. A tumor contains far more than malignant cells. Blood vessels, fibroblasts, immune cells, structural proteins, chemical messengers, extracellular matrix, metabolites, even microorganisms in certain tissues. Together, these components create an ecosystem every bit as dynamic as a rainforest or a coral reef, except this ecosystem favors disease instead of life. Comprehensive research synthesized in journals such as Nature Reviews Cancer and Signal Transduction and Targeted Therapy has revealed that the TME is not an accidental byproduct of tumor growth. It is an actively engineered environment. Cancer reshapes its surroundings to maximize survival. The city is redesigned from the inside out. Consider what happens when any rapidly expanding city consumes more resources than its infrastructure can supply. Traffic slows, garbage accumulates, fresh air becomes scarce, water grows contaminated. The same phenomenon unfolds inside growing tumors. Cancer cells consume extraordinary amounts of glucose. They metabolize nutrients at astonishing speed. Even when oxygen is available, many tumors continue relying heavily on glycolysis, producing large quantities of lactic acid. This altered metabolism gradually acidifies the surrounding environment. Imagine trying to conduct a police investigation inside a swamp filled with corrosive chemicals. Equipment begins failing, communication deteriorates, personnel become fatigued. That is remarkably similar to what immune cells experience. Many white blood cells function poorly in acidic surroundings. Their movement slows, their signaling weakens, their ability to destroy tumor cells declines. Cancer has not merely hidden from investigators, it has poisoned the entire crime scene. But chemistry is only the beginning. Every criminal organization understands another timeless strategy. If you cannot defeat law enforcement, corrupt it. Inside healthy tissues, immune cells cooperate to eliminate threats. Inside tumors, some of those same cells undergo astonishing transformations. Imagine honest police officers gradually accepting bribes, not because they intended corruption, but because every surrounding influence encouraged compromise. Among the most striking examples are macrophages. Normally, macrophages patrol tissues, removing debris, swallowing pathogens, and coordinating immune defense. They are professional cleanup crews, investigators, first responders. Yet, within many tumors, chemical signals reshape their behavior. Instead of attacking cancer, they begin supporting it. These altered cells are often called tumor-associated macrophages. Rather than sounding alarms, they release growth factors, promote blood vessel formation, suppress aggressive immune responses, assist tissue remodeling. The guardians become caretakers of the invader, not through force, through persuasion. Another group enters the conspiracy. Cells known as regulatory T cells. Their normal mission is noble. Prevent excessive immune reactions. Protect healthy tissues from accidental attack. Without them, autoimmune disease could become catastrophic. Cancer recognizes their value immediately. The tumor recruits increasing numbers of these peacekeepers into its territory. Imagine sending conflict mediators into a city already controlled by organized crime. Their calls for restraint, however well-intentioned, benefit the criminals far more than the innocent citizens. Inside tumors, regulatory T cells often suppress the very killer T cells capable of eliminating malignant cells. Again, biology's protective systems become instruments of deception. The conspiracy extends even further. Cancer-associated fibroblasts. Once ordinary structural cells, they begin remodeling the landscape itself. Roads become barricades. Dense networks of extracellular matrix surround the tumor. Imagine detectives attempting to reach a suspect hidden inside a fortress whose streets have been deliberately blocked. Even when immune soldiers know exactly where cancer resides, physical barriers slow their advance. Some never arrive, others become trapped at the edges. The battlefield itself has been redesigned. Then comes perhaps the most disturbing discovery. The toxic chemicals accumulating inside tumors do more than exhaust immune cells. They actively interfere with cellular communication. As cancer cells consume nutrients relentlessly, essential resources become scarce. Glucose disappears, certain amino acids become depleted, oxygen concentrations fall, waste products accumulate. Imagine firefighters entering a burning building carrying empty oxygen tanks. The mission becomes nearly impossible before it begins. Immune cells require energy to move, to divide, to manufacture weapons, to communicate. The tumor microenvironment quietly deprives them of all four. Researchers now understand that metabolic competition forms one of cancer's most effective defense strategies. The tumor feeds first. The immune system survives on whatever remains. Perhaps the greatest illusion surrounding cancer is the belief that tumors simply grow larger over time. Growth is only one part of the story. Transformation is the other. Each passing week, the ecosystem becomes increasingly hostile to immune surveillance. Every new blood vessel, every altered chemical gradient, every recruited suppressive cell, every acidic pocket, every depleted nutrient, together they reshape the battlefield. Eventually, Even highly functional immune cells entering this environment begin behaving differently, not because they forgot their mission, because the city itself changed who they could become. This realization fundamentally altered modern oncology. Destroying cancer cells alone may not always be sufficient. Imagine arresting a crime boss while leaving every corrupt institution untouched. Soon, another leader rises. The same conditions produce the same outcome. Researchers increasingly investigate therapies capable of altering the tumor microenvironment itself, neutralizing acidity, reprogramming macrophages, reducing suppressive immune populations, improving blood vessel function, restoring nutrient availability, breaking down physical barriers. The goal is no longer merely eliminating the criminal, it is rebuilding the city. A healthy ecosystem naturally supports justice. A toxic ecosystem protects corruption. One of the most fascinating conclusions emerging from modern research is that cancer behaves less like a lone assassin and more like the architect of an entire civilization. Every component serves a purpose. Every chemical messenger influences another. Every recruited cell strengthens the network. Like history's great underground organizations, success depends not only upon powerful leaders, but upon countless ordinary participants performing specialized roles. The tumor microenvironment functions as an invisible government operating beneath normal biology. Its laws differ, its priorities differ, its loyalties differ. And until those hidden institutions are understood, the visible tumor reveals only part of the conspiracy. As our investigation closes tonight, Gotham City stands before us in a new light. The tragedy was never simply the existence of criminals. It was the gradual corruption of everything surrounding them. The poisoned streets, the frightened citizens, the compromised officials, the silence replacing justice. Cancer follows the same blueprint. It does not merely hide inside the body. It transforms the body into a place where hiding becomes effortless. The immune system enters expecting a battlefield. Instead, it discovers a city whose every street favors the enemy. Yet every corrupt empire contains a weakness. Every conspiracy leaves traces. Every hidden government depends upon secret supply lines. And in our next investigation, we will begin tracing those invisible networks. Because behind every toxic ecosystem lies an even more astonishing mystery: how cancer communicates across vast distances inside the body, sending encrypted messages that prepare entirely new territories, long before the first invading cell ever arrives. The conspiracy, it seems, extends far beyond the original crime scene, and the map of that hidden network is only beginning to emerge. Part 5. The Recruited Traitors, when the body's guardians switch sides. Every intelligence agency fears one threat above all others: not the foreign spy, not the encrypted message, not even the hidden weapon. The greatest nightmare is the trusted officer who quietly changes allegiance. History is filled with stories of double agents, men and women who once defended their nation only to become its greatest vulnerability. They knew every protocol, every password, every weakness. Once they crossed the line, they became far more dangerous than any outsider. Hollywood has built entire generations of spy films around this idea: the loyal operative, the impossible mission, the shocking revelation that someone inside headquarters has been feeding information to the enemy all along. The audience always asks the same question: why? Were they threatened, blackmailed, ideologically transformed, or simply rewarded? Inside the human body, cancer has mastered precisely this strategy. It rarely defeats the immune system through overwhelming force. Instead, it recruits members from within. Tonight's investigation examines perhaps the darkest chapter yet: not the spies who sneak into the city, but the police officers who begin protecting them. In every healthy tissue, countless immune cells patrol with extraordinary discipline. Some hunt viruses, others destroy bacteria, some repair injuries, others remove dead cells before they become dangerous. Among the most remarkable of these guardians are the macrophages. Their name comes from Greek words meaning big eaters, an appropriate description. Macrophages swallow damaged cells, consume microbes, clean debris, coordinate inflammation, warn neighboring immune cells of danger. Imagine veteran detectives who know every neighborhood. They arrive first at crime scenes, collect evidence, question witnesses, call for reinforcements. Without macrophages, the body's security network would collapse. Yet astonishingly, Many cancers persuade these very detectives to protect the criminals instead. For many years, scientists assumed macrophages represented a single type of immune cell. Research gradually revealed something far more nuanced. Macrophages are extraordinarily adaptable. They change their behavior depending on the environment surrounding them. Imagine a police officer responding to different emergencies. One day, a violent robbery. Another day, a natural disaster. Another, a lost child. The same officer behaves differently because the circumstances demand different responses. Biologists often describe macrophages using two simplified functional states. The first, known as M1, aggressive, inflammatory, highly effective against infections and tumors. These are the detectives pursuing criminals relentlessly. The second, known as M2, calming, repair-oriented, focused on wound healing, reducing inflammation, encouraging tissue rebuilding. Normally, both states serve essential purposes. The body requires warriors, the body also requires healers. Health depends upon balance. Cancer discovered how to exploit that balance. Imagine arriving at a city under attack. The police begin pursuing dangerous criminals. Then suddenly, false reports flood dispatch centers. The crisis is over. Stand down. Focus on rebuilding instead. Repair damaged buildings. Calm the public. Stop making arrests. None of the officers realize the messages originated from the criminals themselves. This resembles what happens inside many tumors. Cancer cells release an astonishing collection of chemical messengers, cytokines, growth factors, metabolic signals, extracellular vesicles. Each carries subtle instructions influencing nearby immune cells. Over time, these signals persuade macrophages to abandon their aggressive M1 behavior. Instead, they gradually adopt characteristics resembling M2 macrophages. Scientists commonly refer to these converted cells as tumor-associated macrophages, or TAMs. Their transformation changes everything. A detective who stops investigating is troubling. A detective who begins assisting criminals is catastrophic. Tumor-associated macrophages often release molecules that encourage blood vessel formation. The tumor receives fresh oxygen, fresh nutrients, fresh escape routes. They produce enzymes capable of remodeling surrounding tissues. Physical barriers dissolve. Cancer spreads more easily. They suppress killer T cells, reduce inflammatory signals, recruit additional suppressive immune populations. Instead of eliminating malignant cells, they help create conditions favorable to tumor survival. Imagine police officers directing traffic, not away from organized crime, but toward it. The city still sees uniforms. The badges remain authentic. Yet the loyalty has shifted. How could such a profound change occur? Was it merely temporary influence or something deeper? Modern research increasingly points toward an extraordinary mechanism, epigenetic reprogramming, to appreciate its significance. Imagine discovering that a trusted intelligence officer has not simply received different orders. His training manuals have been rewritten, his communication protocols updated, his instincts gradually altered. Not his identity, his programming. Genes alone do not determine cellular behavior. Cells also regulate which genes remain active and which remain silent. These epigenetic controls function like bookmarks inside an enormous instruction manual. The text remains unchanged, but different chapters become accessible. Research from investigators at Harvard Medical School, along with many collaborators worldwide, has shown how tumors can reshape these regulatory programs inside macrophages. Signals from the tumor alter epigenetic patterns. Genes promoting aggressive anti-tumor activity become quieter. Genes supporting tissue repair and immune suppression become increasingly active. The detective still exists, but his priorities have changed. The conspiracy extends even further. Imagine organized crime establishing an academy dedicated to recruiting entirely new collaborators, not seasoned detectives, young recruits, individuals whose loyalties remain uncertain. Cancer accomplishes something remarkably similar through another population of immune cells called myeloid-derived suppressor cells, better known as MDSCs. Under healthy conditions, immature myeloid cells mature into useful immune defenders, macrophages, granulocytes, other protective cells. Cancer interrupts that developmental journey. Instead, large numbers of immature suppressive cells accumulate. Rather than fighting disease, they actively weaken immune responses. Picture military cadets intercepted before graduation, trained by enemy instructors, then deployed against their own nation. MDSCs interfere with killer T cells, consume nutrients essential for immune activity, produce suppressive chemicals, promote tumor growth, and assist the creation of the toxic ecosystem we explored in our previous investigation. Cancer has not merely corrupted experienced officers, it has begun recruiting the next generation. Hollywood often portrays betrayal as a dramatic moment, one shocking revelation, one decisive choice. Biology is rarely so theatrical. Macrophages do not wake one morning and consciously choose corruption. Their transformation unfolds gradually. Thousands of molecular conversations, thousands of chemical signals, tiny changes accumulating day after day. Each message seems insignificant. Together, they rewrite an entire identity. Cancer behaves less like a dictator issuing commands and more like an extraordinarily persuasive propagandist. Every surrounding influence encourages compromise. Eventually, doing the wrong thing feels completely natural. Perhaps the most unsettling discovery is that these recruited immune cells often become essential components of tumor survival. Remove them experimentally, and some tumors shrink dramatically. Reprogram them back toward anti-tumor activity, and immune responses strengthen. This realization has inspired an entirely new direction in cancer therapy. Instead of asking, how do we destroy macrophages? Researchers increasingly ask, how do we remind them who they were? Can epigenetic programming be reversed? Can suppressive macrophages recover their original identity? Can MDSCs be prevented from accumulating? Can loyal officers be rescued before corruption becomes permanent? These questions now occupy laboratories across the world. The investigation reveals another profound lesson. Cancer does not simply evolve genetically, it evolves socially. It builds alliances, forms partnerships, manipulates relationships. Every recruited macrophage becomes another political victory. Every suppressive immune cell expands the conspiracy. The tumor succeeds not because every surrounding cell becomes malignant, but because enough normal cells begin serving malignant interests. History repeatedly demonstrates this principle. Power rarely depends upon numbers alone. It depends upon influence. Consider again the spy thriller. The audience usually imagines the double agent acting for personal gain. Money, power, ideology. Inside biology, the motivations differ entirely. Macrophages are not evil. MDSCs are not malicious. They respond to information, change behavior according to signals, perform exactly what their environment instructs. Cancer's genius lies not in creating monsters. It lies in controlling the information those cells receive. The message changes, the mission changes, the outcome changes. As tonight's case file closes, we uncover perhaps the most disturbing revelation yet. The tumor does not stand alone inside the body. It commands an expanding network of converted allies, former guardians, former investigators, former protectors, each wearing the same biological uniform they always possessed. From the outside, nothing appears different, yet beneath the surface, their loyalty belongs elsewhere. The greatest conspiracies in history were never built solely by villains. They survived because ordinary institutions gradually stopped resisting them. Cancer follows the same ancient blueprint. It corrupts before it conquers, it persuades before it destroys, and by the time the betrayal becomes visible, the organization protecting it has already grown remarkably strong. But every conspiracy leaves records. Every covert network depends upon hidden communication. In our next investigation, we will begin intercepting those secret transmissions. Because cancer is not merely recruiting accomplices nearby, it is sending encrypted biological messages across the body, preparing distant territories for an invasion that has not yet begun. The operation is larger than anyone first imagined, and the next clue is already waiting. Part 6. The War for Energy. The Warburg Effect and the Starvation of the Immune Army. Every siege in history has followed a familiar pattern. The winning army does not always possess the largest number of soldiers. Sometimes it simply controls the food. Imagine an American city under siege. The roads remain open, but only for one side. Warehouses of grain disappear overnight. Fuel depots are seized. Water purification plants are occupied. Hospitals begin rationing medicine. Ordinary citizens are not defeated by bullets. They are defeated by hunger. The defenders still have weapons. They still have courage. They still have trained officers. But courage burns calories. Weapons require fuel. Without resources, the strongest army eventually becomes powerless. Cancer discovered this ancient military strategy long before humans ever wrote it into history books. It does not merely fight the immune system, it starves it. Tonight, our investigation enters one of the most fascinating battlegrounds in modern biology. Not genes, not disguises, not secret passwords, energy. Because every war, whether fought between nations or between cells, is ultimately won by whoever controls the supply lines. Earlier, we uncovered how tumors corrupted immune cells, poisoned their surroundings, and silenced molecular alarms. Yet investigators continued noticing another mystery. Even when killer T cells successfully entered tumors, many slowed down, their attacks weakened. Some stopped dividing, others simply disappeared. At first glance, they resembled exhausted soldiers. The obvious explanation was fatigue. The real explanation proved even more disturbing. They were starving. Every living cell obeys the same universal law. No energy, no function. Muscles cannot contract, brains cannot think, hearts cannot beat, immune cells cannot hunt. The primary fuel for many cells is glucose, a simple sugar. Yet inside the human body, glucose represents something far, far greater than nutrition. It is biological currency, the fuel that powers nearly every critical operation. Imagine an entire city depending upon electricity. If one organization secretly monopolizes every power station, every factory, every fuel pipeline, then everyone else becomes dependent upon its decisions. Cancer behaves in remarkably similar fashion. It becomes an aggressive economic monopolist. Nearly a century ago, a brilliant German physician and scientist noticed something profoundly unusual. Even when oxygen was plentiful, many cancer cells continued breaking down glucose through a relatively inefficient process called glycolysis. Ordinarily, healthy cells prefer extracting energy through oxidative phosphorylation inside mitochondria when oxygen is available. That pathway generates far more energy from each glucose molecule. Cancer, however, often chooses differently. It consumes glucose at astonishing speed while relying heavily on aerobic glycolysis. Today, this phenomenon is known as the Warburg effect. At first, scientists considered it wasteful. Why would rapidly growing cells deliberately choose a less efficient strategy? The answer emerged gradually. Efficiency was never the objective. Speed was. Imagine two factories. One produces products slowly but extracts maximum value from every shipment. The other wastes raw materials, yet manufactures goods at extraordinary speed. For a city expanding uncontrollably, speed matters more than efficiency. Cancer adopted the same philosophy. The consequences extend far beyond the tumor itself. Because cancer consumes enormous amounts of glucose, less remains available for neighboring immune cells. Imagine emergency responders arriving after a disaster, only to discover that every fuel station has already been emptied. The fire engines still exist. The rescue equipment remains intact, but nothing powers them. Killer T cells require glucose for activation, for movement, for division, for manufacturing the molecular weapons used to destroy infected or malignant cells. Without adequate energy, their effectiveness declines dramatically. The soldiers are present. The ammunition exists. The fuel is gone. Cancer has transformed the battlefield into an economic blockade. Yet monopolizing glucose represents only half the operation. Every industrial process produces waste. The Warburg effect generates large quantities of lactic acid. As tumors expand, this acid accumulates throughout the surrounding environment. Imagine rivers flowing through a city becoming increasingly contaminated. Factories continue operating. Citizens remain alive, but every breath becomes more difficult. Every movement requires greater effort. Inside tumors, the growing concentration of acid alters the chemistry of the entire battlefield. Immune cells struggle under these conditions. Their communication weakens, their ability to release toxic molecules declines, their movement slows. The city itself becomes hostile to those attempting to defend it. Picture an elite tactical unit attempting to pursue dangerous fugitives through knee-deep swamp water. Every step demands enormous effort. Radio communication fades, visibility deteriorates. The fugitives know every shortcut, the officers do not. This captures the challenge faced by killer T cells inside acidic tumors. They possess extraordinary training, remarkable precision, powerful biological weapons, yet the environment steadily erodes every advantage. The problem is no longer courage, it is terrain. Another invisible accomplice strengthens the conspiracy. As tumors consume oxygen rapidly while growing faster than blood vessels can adequately supply it, regions of hypoxia begin forming, areas where oxygen becomes scarce. Imagine neighborhoods plunged into darkness after every electrical station has been captured. Hospitals lose power. Traffic lights fail. Communication towers collapse. Everything slows. Hypoxia profoundly alters cellular behavior. Cancer cells activate survival programs. New blood vessels begin forming. Additional suppressive signals spread throughout the tissue. Meanwhile, immune cells face increasingly hostile conditions. Research into metabolic restructuring, including studies published through platforms such as Science Direct and in journals including Molecular Immunology, has demonstrated how hypoxic acidic environments significantly impair T cell migration and function. The investigators are no longer merely underfunded. They are operating inside a city deprived of light, oxygen, and fuel. Consider what movement truly requires. Every step taken by a T cell demands energy. Every change in direction, every encounter with a target, every molecular weapon released. Energy fuels all of it. When glucose disappears, oxygen declines, and acid accumulates, movement itself becomes increasingly difficult. Imagine detectives pursuing a suspect across a collapsing bridge while carrying empty oxygen tanks. Even locating the criminal becomes an extraordinary challenge. Cancer does not necessarily defeat immune cells through superior weapons. It exhausts them through relentless logistics. History repeatedly confirms this principle. Great armies have collapsed without losing decisive battles. Supply lines failed first. The brilliance of this strategy lies in its subtlety. Cancer does not issue commands saying, destroy the immune system. Instead, it quietly changes the economic landscape. Resources become scarce. Waste products accumulate, environmental conditions deteriorate. Every individual obstacle appears manageable. Together, they create a crisis impossible to ignore. This is precisely how many urban wars unfold, not through one catastrophic explosion, but through gradual shortages, fuel today, food tomorrow, medicine next week. Eventually, the city simply cannot resist. Modern cancer research increasingly recognizes metabolism as one of the central battlefields of oncology. Scientists once viewed metabolism merely as housekeeping, a background process. Now, it appears more like military logistics: control the fuel, control the war. Investigators explore therapies designed to interrupt cancer's metabolic advantage. Some aim to reduce excessive glucose consumption. Others seek to neutralize acidic conditions. Still others attempt to improve oxygen delivery or restore metabolic flexibility to exhausted immune cells. The objective is no longer only killing tumors. It is rebuilding fair access to essential resources. Because an immune army cannot defend a nation if every supply convoy has already been captured. Perhaps the most fascinating lesson emerging from this investigation is that biology resembles economics far more than many once imagined. Every cell competes, negotiates, consumes, produces, invests, adapts. The healthiest tissues maintain remarkable balance. No single population monopolizes everything. Cancer breaks that balance. It transforms shared resources into private property. The tumor prospers. Everyone else pays the price. History teaches that monopolies eventually reshape entire civilizations. Once essential goods become controlled by a single power, freedom gradually disappears. The same pattern unfolds inside the tumor microenvironment. Glucose belongs to the tumor. Oxygen becomes scarce. Acid dominates the streets. The immune system still possesses extraordinary weapons, yet increasingly lacks the energy to use them. The conspiracy succeeds not because cancer invented stronger soldiers, it seized the economy. As tonight's investigation concludes, one truth stands above all others. Every detective knows that solving a crime requires more than identifying the suspect. The investigators themselves must remain fed, equipped, rested, supported. Cancer understood this principle long before modern medicine did. Rather than confronting the immune system head-on, it quietly captured the supply chains, the warehouses, the fuel depots, the transportation routes. By the time the defenders recognized the trap, their reserves were already running dangerously low. But every monopoly contains vulnerabilities. Every supply network has hidden intersections. And in our next chapter, we will uncover perhaps the most sophisticated communication system yet discovered. Because cancer is not merely competing for energy. It is broadcasting encrypted biochemical messages across the body, sending microscopic packages filled with instructions, preparing distant landscapes long before the first metastatic cell ever arrives. The siege, it turns out, was only the opening move. The real operation has already spread far beyond the original battlefield. Part 7. The Energy Thief. The high-tech heist of stolen mitochondria. Every era has produced a crime so unexpected that investigators initially refused to believe it. There was a time when stealing money meant robbing a bank. Then came cybercrime. Hackers no longer carried guns, they stole electricity without touching a power plant, they emptied bank accounts without entering a vault, they infiltrated critical infrastructure from thousands of miles away. The battlefield had changed. Now imagine something even more astonishing. Imagine a cybercriminal who does not merely steal electricity from your home. He hacks directly into your personal generator, disconnects it, and installs it inside his own building. Your lights fade. His become brighter. You are weakened. He becomes stronger. It sounds like science fiction. Yet modern cancer biology has uncovered evidence suggesting that something remarkably similar may occur inside the human body. Not through wires, not through software, but through living cells. Tonight's investigation explores one of the newest and most astonishing discoveries in tumor biology, a crime so sophisticated it rewrites our understanding of how cancer competes with the immune system. Until recently, we believed tumors starved immune cells. Now, there is evidence they may do something even more audacious. They may steal their power plants. Our investigation has already exposed a long chain of conspiracies. Cancer erased its identity cards. It forged biological stop signals. It corrupted immune officers. It poisoned entire neighborhoods. It monopolized energy supplies. Every chapter revealed a deeper level of deception, yet one mystery remained unresolved. Researchers repeatedly observed tumor-infiltrating lymphocytes, the elite T cells that successfully penetrated deep into cancers. These soldiers had overcome extraordinary obstacles. They crossed physical barriers, ignored suppressive signals, survived acidic environments, reached the enemy itself, and still, many suddenly lost their strength. Not gradually, sometimes astonishingly quickly. It resembled an army whose batteries had vanished overnight. Where had the energy gone? To answer that question, we must revisit one of biology's greatest inventions, the mitochondrion, often called the powerhouse of the cell. The comparison is accurate but incomplete. Imagine every city operating thousands of miniature power stations. Electricity flows continuously, factories remain productive, communication networks function, transportation continues. Remove the power stations, and the city still exists. But little actually works. Mitochondria perform a remarkably similar role. They convert nutrients into usable cellular energy, powering movement, repair, communication, protein production, cell survival, even decisions about life and death. For immune cells, healthy mitochondria are especially important. A killer T cell cannot pursue cancer without enormous energy demands. Every movement, every division, every attack, every memory response depends upon mitochondrial performance. A tired immune cell often possesses tired mitochondria. But what if fatigue were not the whole story? What if someone had physically removed the generators? At first, the very idea sounded impossible. Cells exchange chemical messages constantly. Hormones, proteins, small molecules, even fragments of genetic material. But entire mitochondria? Living organelles? Transferred from one cell to another? It challenged decades of biological assumptions. Gradually, evidence accumulated. Researchers began documenting rare but undeniable examples of mitochondrial transfer. Healthy cells could donate mitochondria during tissue repair. Injured cells sometimes recovered after receiving them. Nature already possessed mechanisms allowing this remarkable exchange. The discovery inspired optimism. Perhaps damaged organs could someday be healed through mitochondrial donation. Then investigators asked a far darker question. If healthy cells can transfer mitochondria, could cancer exploit the same process? Imagine investigators discovering an advanced cybercriminal using emergency rescue technology, not to save victims, but to steal from them. The innovation itself is not evil. Its purpose has been reversed. Cancer appears to follow the same blueprint. Microscopic structures called tunneling nanotubes began attracting scientific attention. These astonishing biological bridges connect neighboring cells through extremely thin membrane channels. Picture secret fiber optic cables stretching between buildings, invisible, temporary, yet fully functional. Through these tiny tunnels, cells can exchange cargo, proteins, signaling molecules, even entire organelles under certain conditions. The discovery was extraordinary. The implications, even more so. Then came one of the most startling findings in modern tumor immunology. Research published in Nature reported evidence suggesting that within tumors, mitochondria could be transferred from tumor-infiltrating lymphocytes, often called T-hills, into neighboring cancer cells. Pause for a moment. Consider what this means. The immune soldier arrives carrying fully functional energy generators. The cancer cell, already competing aggressively for survival, acquires those generators. One cell weakens, the other grows stronger. The battlefield shifts before a single molecular weapon is fired. Imagine a military convoy entering hostile territory. Instead of merely losing fuel, the enemy steals the engines themselves. The vehicles remain, the drivers remain, but the machinery powering the mission disappears. Why would such transfer occur? Researchers continue investigating the precise mechanisms. Many questions remain unanswered. Biology rarely offers simple explanations, yet the emerging evidence paints a fascinating picture. Within the hostile tumor microenvironment, immune cells experience profound stress, low oxygen, high acidity, nutrient deprivation, continuous activation. Under these extraordinary conditions, cellular communication pathways change dramatically. Tunneling nanotubes may become increasingly important, whether initiated by immune cells under stress, by tumor cells, or through complex bidirectional signaling, likely depends on context that scientists are still working to understand. What is becoming increasingly clear is that cancer possesses an astonishing ability to exploit nearly every biological system available. If mitochondria become transferable resources, the tumor appears capable of benefiting from that exchange. Imagine the psychological impact upon an intelligence agency discovering that every encrypted laptop issued to field agents could secretly be commandeered by enemy hackers. The agents would still possess information, training, determination. Yet every operation would begin at a profound disadvantage. This mirrors what happens to exhausted tumor-infiltrating lymphocytes. Without robust mitochondrial function, their movement slows, their persistence declines, their ability to multiply weakens. Even when recognizing cancer perfectly, they may lack sufficient energy to complete the mission. The detective has solved the case. The police car no longer starts. Meanwhile, the tumor enjoys an extraordinary advantage. Additional mitochondria may support metabolic flexibility, energy production, survival under stressful conditions. The invader becomes increasingly resilient precisely as the defender becomes increasingly exhausted. Every successful transfer deepens the imbalance, not through superior genetics alone, through captured infrastructure. History teaches that conquering nations often seize railroads, power plants, communications networks, rather than rebuilding them from nothing. Cancer appears to follow the same strategic principle. Why construct new resources if neighboring cells already possess them? This discovery transforms our understanding of competition inside tumors. For many years, scientists described cancer primarily as a disease of uncontrolled growth. Today, another picture emerges. Cancer resembles an adaptive ecosystem engaged in continuous resource acquisition. Genes, immune signals, blood vessels, nutrients, oxygen, structural support, now perhaps even mitochondria. Nothing useful remains beyond negotiation or exploitation. Every nearby cell becomes both neighbor and potential supplier. The implications for future medicine are profound. If mitochondrial transfer contributes meaningfully to immune exhaustion, could blocking tunneling nanotubes strengthen anti-tumor immunity? Could therapies preserve mitochondrial integrity inside killer T cells? Could engineered immune cells resist energy theft? These questions now occupy laboratories around the world. Researchers recognize that restoring immune strength may involve more than activating T cells. Their energy infrastructure must also remain protected. A soldier without ammunition cannot fight. A soldier without electricity cannot even reach the battlefield. Perhaps the most unsettling lesson from tonight's investigation is philosophical rather than medical. Throughout history, the most sophisticated crimes rarely relied upon brute force. They exploited existing systems, banking networks, telegraph lines, railroads, computers, the internet. Cancer demonstrates exactly the same evolutionary genius. It rarely invents entirely new biology. Instead, it repurposes biology already present. Repair mechanisms become escape routes. Protective checkpoints become counterfeit credentials. Communication systems become propaganda channels. And now, even cellular cooperation may become an opportunity for theft. As we close another encrypted case file, one truth grows impossible to ignore. Cancer is no ordinary adversary. It behaves less like a reckless invader and more like an elite intelligence organization. Patient, adaptive, resourceful, always searching for overlooked vulnerabilities. The immune system brings courage, precision, remarkable technology shaped by hundreds of millions of years of evolution. Cancer answers with deception, not because deception is stronger than force, but because deception changes the battlefield before force can even be applied. The greatest hackers rarely destroy computers, they quietly gain administrator privileges. Cancer appears to have mastered an equally astonishing trick. Rather than merely starving its enemies, it may appropriate the very engines that keep them alive. Yet every sophisticated cyber attack leaves digital fingerprints. Every hidden tunnel can eventually be mapped. Every stolen power station can be traced back to its source. And in our next investigation, we will uncover another layer of this biological conspiracy: how cancer sends microscopic encrypted packages throughout the bloodstream, preparing distant organs for invasion long before the first metastatic cell ever arrives. The theft of energy was never the final objective. It was preparation for something far larger. The next operation has already begun. Part 8, the Fibrotic Shield, the impenetrable fortress of stone and steel. Every great fortress in history shared one simple truth. The strongest defense was never the soldiers standing on the walls, it was the walls themselves. Imagine a massive wartime bunker buried beneath reinforced concrete. Steel doors several feet thick, layers of barbed wire stretching for miles, anti-tank obstacles scattered across the surrounding fields, watchtowers scanning every approach. Even the most highly trained Special Forces unit cannot accomplish its mission if it never reaches the entrance. Victory is impossible because access is impossible. For centuries, military engineers understood this principle. Do not merely strengthen the defenders, prevent the enemy from arriving. Cancer learned the same lesson. It discovered that defeating the immune system is not always necessary. Sometimes, keeping the immune system outside is enough. Tonight, our investigation uncovers another extraordinary layer of the conspiracy. Not deception, not starvation, not corruption, architecture. Because the most successful criminal organizations eventually stop hiding. Instead, they build fortresses no detective can enter. Our previous investigations revealed a remarkable sequence of biological espionage. Cancer erased identification badges, forged molecular passwords, bribed immune guardians, captured energy supplies, even stole cellular power plants. Each strategy weakened the body's defenses, yet physicians continued observing an unsettling mystery. Some tumors contained remarkably few immune cells. Not weakened immune cells, not exhausted ones, simply none. It was as though the immune army had never arrived. Where had they gone? Were they avoiding the battlefield, or had the battlefield become unreachable? Imagine studying satellite photographs of a heavily defended military installation. The roads leading toward it have vanished, bridges have been demolished, concrete barriers stretch across every entrance, minefields cover the surrounding terrain. Reconnaissance confirms thousands of elite soldiers remain outside. Ready, prepared, determined, yet none can penetrate the perimeter. This is strikingly similar to what modern scientists have discovered inside many human cancers. The obstacle is not always biological recognition. Sometimes it is simple physical exclusion. Cancer rarely constructs this fortress alone. Every major empire relies upon engineers, architects, construction crews. Inside tumors, those builders are often cancer-associated fibroblasts, commonly known as CAFs. Under healthy conditions, fibroblasts perform honorable work. They repair injured tissues, produce structural proteins, help wounds heal, restore damaged organs. Imagine skilled construction workers rebuilding a city after an earthquake. Without them, recovery would be impossible. Cancer does not destroy these builders. It recruits them. The workers receive new instructions. The mission changes. Instead of rebuilding healthy neighborhoods, they begin constructing defensive fortifications around the tumor itself. The primary building material is something called the extracellular matrix, often abbreviated the ECM. To appreciate its importance, imagine the framework supporting an entire city: roads, bridges, scaffolding, foundations, pavement, everything connecting individual buildings into one functioning environment. Healthy tissues require extracellular matrix. Without it, organs lose structure, cells lose organization, life becomes impossible. Cancer exploits this essential infrastructure. Activated CAFs begin producing enormous quantities of collagen, fibronectin, and other structural proteins. Layer upon layer, fiber upon fiber, the surrounding tissue gradually thickens, hardens, compacts. The tumor becomes enclosed within a dense biological fortress. The prison walls are built, not by the criminals, but by ordinary citizens persuaded they are performing necessary repairs. Imagine driving toward an emergency rescue mission. Every highway is blocked by concrete barricades. Every tunnel has collapsed. Every bridge has been replaced with tangled steel. The rescue team still possesses extraordinary equipment. They simply cannot reach the victims. Killer T cells experience an astonishingly similar problem. Even when they recognize cancer perfectly, even when molecular disguises fail, even when checkpoint inhibitors reactivate their weapons, many still cannot physically enter the deepest regions of the tumor. The extracellular matrix becomes an enormous mechanical obstacle, not invisible, not mysterious, simply overwhelming. The immune army stands outside the walls. The battle never begins. Researchers at the U.S. National Cancer Institute and many collaborating institutions have carefully documented these physical barriers within numerous cancers. The evidence reshaped one of oncology's fundamental assumptions. For years, scientists concentrated primarily on molecular communication, signals, genes, proteins, receptors. Those remain critically important. Yet equally significant is the simple geography of the battlefield. Who can reach whom? Which roads remain open? Which pathways have collapsed? Sometimes distance alone determines survival. One particularly important concept emerging from this work is the idea of the cold tumor. The phrase sounds almost poetic. Its meaning is profoundly practical. A hot tumor contains abundant immune cells. The soldiers have arrived. The fighting has begun. A cold tumor, by contrast, contains remarkably few functional immune cells within its core. Not because the immune system never responded, often because immune cells were excluded before they could enter. Imagine a fortress surrounded by thousands of allied troops. The army exists. The mission exists. The gates never open. Cold tumors represent one of the greatest challenges facing modern immunotherapy. Even highly effective immune soldiers cannot eliminate enemies hidden behind impenetrable walls. The fortress possesses another advantage. Dense extracellular matrix compresses nearby blood vessels. Imagine supply trucks attempting to reach a besieged city through roads narrowed by collapsing buildings. Traffic slows, deliveries decline. Inside tumors, compressed blood vessels reduce oxygen delivery, limit nutrient exchange, restrict immune cell trafficking. Ironically, the fortress protecting cancer simultaneously worsens hypoxia, the oxygen shortage we explored in our previous investigation. Every defensive strategy strengthens another. The conspiracy becomes self-reinforcing. Thicker walls create poorer circulation. Poorer circulation strengthens immune suppression. Immune suppression permits further construction. The cycle continues. Military historians often describe defense in layers. First, barbed wire, then minefields, then concrete barriers, then defensive bunkers, finally, armed defenders. Cancer appears to follow remarkably similar logic. First, the immune system struggles to recognize the tumor. Then, checkpoint signals weaken attacking cells. Then, toxic metabolism drains their energy. Then, corrupted immune allies reinforce suppression. Finally, physical architecture prevents entry altogether. Each defensive layer compensates for weaknesses in another. No fortress depends upon one wall. The entire system functions together. Perhaps the most fascinating discovery is that cancer-associated fibroblasts do far more than construct physical barriers. They also produce chemical signals, recruit suppressive immune cells, influence blood vessel formation, modify tissue stiffness, communicate continuously with neighboring cancer cells. The architects are also intelligence officers. Construction workers become strategic planners. The fortress evolves while the battle continues. Every new threat triggers further adaptation. Modern cancer research increasingly investigates methods for dismantling these defensive structures. Could dense extracellular matrix be remodeled? Could CAF activity be redirected? Could immune cells receive alternative routes into the tumor? Could blood vessels be normalized? These questions have become central to contemporary oncology. The objective resembles military engineering. Sometimes, victory depends less upon stronger soldiers than upon removing the walls blocking their advance. An elite rescue team requires access before skill can matter. History repeatedly demonstrates that no fortress remains invincible forever. Ancient cities, once believed unconquerable, eventually fell, sometimes through superior engineering, sometimes through hidden tunnels, sometimes because supply lines collapsed, sometimes because defenders opened the gates themselves. Every wall contains weaknesses. The challenge lies in finding them. Cancer's fortress appears formidable, yet scientists increasingly map its architecture with extraordinary precision. Every collagen fiber, every fibroblast, every compressed blood vessel, every excluded immune cell becomes another clue. The blueprint is gradually emerging. As tonight's investigation draws to a close, the picture before us has become unmistakable. Cancer is not merely a disease of uncontrolled growth. It is an architect of defensive civilizations. It constructs barriers, controls geography, shapes traffic, rebuilds landscapes. The immune system does not always lose because it lacks courage. Sometimes it never receives permission to enter the city. The battlefield itself has been redesigned to guarantee failure before combat begins. The strongest army in history remains powerless if trapped outside the gates. Yet history also teaches another lesson. Every fortress eventually reveals a hidden entrance, a forgotten passage, a structural weakness overlooked by its builders. And in our next investigation, we will pursue an even stranger mystery. Because cancer's greatest achievement may not be building walls around itself, but sending encrypted biological couriers far beyond those walls. Microscopic messengers that travel through the bloodstream, preparing distant organs long before invasion begins. The fortress, we will discover, was never meant merely for defense. It was headquarters for a much larger operation. And the next chapter will reveal the first evidence of that hidden network. Haber Part 9, The Mastermind's Game, The Hidden World of Cancer Stem Cells. Every great criminal organization eventually leaves investigators with the same haunting question. How can the syndicate survive after every visible leader has been captured? The warehouses have been raided, the foot soldiers arrested, the money seized, the headquarters destroyed. Newspapers declare victory. Politicians congratulate the investigators. Citizens celebrate in the streets. Then, months later, the organization returns, new recruits, new operations, new leadership, almost as though someone had quietly rebuilt the entire empire from the shadows. History has repeated this mystery countless times. The most dangerous criminal is rarely the one standing in public view. The true architect seldom appears at the crime scene. He rarely carries a weapon. He rarely gives orders in person. He remains hidden, patient, invisible. If necessary, he sacrifices hundreds of followers to protect his own identity. As long as he survives, the organization survives. Cancer appears to possess a remarkably similar figure, a hidden architect, a biological mastermind. Tonight, our investigation reaches one of the deepest conspiracies in modern oncology, the mystery of the cancer stem cell. By now, we have uncovered an extraordinary network of deception. Cancer erased its identity, forged immune passwords, corrupted defenders, poisoned its environment, monopolized nutrients, stole mitochondria, built physical fortresses. Each discovery answered one question while creating another. Why do some cancers return after apparently successful treatment? Patients often hear encouraging news. The tumor has shrunk dramatically. Medical imaging reveals remarkable improvement. Laboratory measurements decline. Everything suggests victory. Then, months or years later, the disease reappears, sometimes in the same location, sometimes elsewhere. How can an empire return after its visible army has been destroyed? Imagine federal investigators dismantling an enormous organized crime network, hundreds of arrests, millions of dollars confiscated, every known safe house eliminated. Only later do they discover one overlooked individual, the original strategist, hidden under a false identity, living quietly, never communicating directly, never participating in visible crimes. When conditions become favorable again, he recruits new followers, builds new headquarters, restores the organization. The previous operation, despite enormous success, never truly ended. Many scientists now believe certain cancers behave in a surprisingly similar way. Within the tumor, a relatively small population of cells may possess extraordinary abilities. These are called cancer stem cells, or CSCs. The word stem does not imply that they originated from normal stem cells in every case. Rather, it reflects what they can do. They can renew themselves, generate diverse tumor cell populations, and potentially rebuild much of the tumor after surviving treatment. To appreciate why this matters, consider how ordinary tumor cells behave. Many divide rapidly, consume energy aggressively, expand the visible mass. These cells form most of what physicians detect through imaging. Chemotherapy often targets precisely this characteristic. Rapid division. Many cancer cells die. The tumor shrinks. The visible army collapses. Yet cancer stem cells frequently behave differently. Some divide slowly. Others remain in a relatively quiet state, almost dormant. Imagine soldiers searching an abandoned headquarters while the true commander waits silently inside an underground bunker. The attack succeeds against the wrong target. This possibility fundamentally altered how researchers think about relapse. Perhaps recurrence does not require every cancer cell to survive. Perhaps only a tiny population must remain, like seeds surviving after a forest fire. The trees disappear, the roots endure. When rain returns, the forest grows again. Cancer stem cells may serve a remarkably similar role, not the majority, but the source. Yet survival alone does not explain their extraordinary resilience. Investigators began asking another question. Why do immune cells struggle to eliminate these hidden architects? Once again, the answer pointed toward deception. Research published through PubMed Central and numerous collaborating laboratories has explored how cancer stem cells display remarkable immune evasive properties. Among the molecules attracting significant attention is CD276. Also known as B7H3, this protein has been associated in many cancers with immune suppression and poor clinical outcomes. Imagine a criminal mastermind surrounding himself with layers of legal protection. Every investigator approaching encounters another obstacle, another false document, another misleading trail. CD276 appears to contribute to an environment where immune attack becomes increasingly difficult. The mastermind remains hidden, while others absorb the attention. Another intriguing marker frequently associated with cancer stem cells is ALDH1. This enzyme participates in important metabolic processes. Yet elevated ALDH1 activity has also become a valuable clue for identifying stem-like populations in numerous tumors. Imagine detectives discovering that members of an elite underground organization consistently carry an unusual encrypted communication device. Not proof of guilt, but a recurring signature. ALDH1 serves a somewhat comparable role. Researchers often use it to identify populations possessing stem-like characteristics, including enhanced resistance to stress and, in many settings, greater therapeutic persistence. The clues continue pointing toward the same conclusion. The hidden architect possesses specialized equipment unavailable to ordinary followers. But perhaps the greatest weapon of cancer stem cells is neither CD276 nor ALDH1. It is adaptability. Imagine interrogating hundreds of captured criminals. Every answer differs. Every identity changes. Every communication route shifts before investigators arrive. Cancer stem cells appear remarkably flexible. They respond to environmental pressures, therapies, immune attacks, metabolic stress. Some may transition between more stem-like and more differentiated states, depending on conditions. The conspiracy does not merely hide, it evolves while hiding. Every failed strategy becomes another lesson. Every surviving cell carries forward the next generation of deception. The immune system faces an extraordinary dilemma. Ordinary tumor cells are numerous, visible, metabolically active. Cancer stem cells may represent only a small fraction of the total population. Finding them resembles searching for one anonymous intelligence officer inside a city of millions. The visible gang attracts headlines. The invisible strategist determines the future. Destroying 99% of the organization means little if the remaining 1% can recruit replacements indefinitely. History repeatedly demonstrates that organizations survive because of leadership. Armies recover after defeat. Corporations rebuild after bankruptcy. Political movements return after suppression. Not because every participant survived, but because the guiding structure endured. Cancer stem cells may fulfill a comparable biological function, not commanding consciously, not issuing deliberate orders, yet serving as reservoirs capable of regenerating complexity after catastrophe. The organization begins again. This realization has profoundly influenced modern cancer research. Scientists increasingly ask questions unimaginable only decades ago. How can cancer stem cells be identified more accurately? Which molecular pathways allow self-renewal? Can immune cells be redirected towards stem-like populations? Can therapies eliminate both rapidly dividing tumor cells and the quieter reservoir capable of rebuilding them? The challenge resembles dismantling organized crime completely. Capturing street-level operatives alone never ends the conspiracy. The architect must also be found. Another fascinating insight emerges from this investigation. Cancer does not depend upon one perfect disguise. It layers protections. If checkpoint molecules fail, the acidic microenvironment remains. If metabolic competition weakens, physical barriers persist. If ordinary tumor cells die, cancer stem cells may survive. Every defense supports another. Every weakness receives reinforcement. Like history's most resilient underground networks, redundancy becomes the secret of survival. Yet investigators continue making progress. Single-cell sequencing reveals unprecedented detail, advanced imaging uncovers hidden cellular neighborhoods. Artificial intelligence analyzes patterns invisible to human observers. New immunotherapies seek increasingly precise targets. The conspiracy remains formidable, but its map grows clearer each year. Every discovery removes another layer of mystery. Every experiment illuminates another corridor inside the fortress. The hidden architect no longer enjoys complete anonymity. As tonight's case file closes, we arrive at perhaps the most unsettling realization of all. The greatest threat inside the tumor may never have been the countless visible cells dominating medical scans. The true danger may have remained quietly concealed from the beginning, watching, waiting, preserving enough biological knowledge to rebuild everything once the storm passed. The world's greatest detectives know that solving individual crimes is never enough. The mastermind must also be found. Cancer appears to follow precisely the same rule. Its visible empire matters. Its hidden architect matters more. But even the most careful mastermind eventually leaves evidence: a financial record, a forgotten message, a trusted courier, a pattern repeated one time too many. And in our next investigation, we will uncover perhaps the most sophisticated communication network in the entire conspiracy. The microscopic encrypted packages known as extracellular vesicles and exosomes, biological couriers carrying instructions across the bloodstream to prepare distant organs for future invasion. Because the hidden mastermind is not merely rebuilding today's organization, it is already planning tomorrow's expansion. Part 10. Evolution Under Fire. How cancer learns to escape every new trap. Every detective eventually faces the same unsettling realization. The suspect is watching the investigation. Every raid changes the criminal's behavior. Every arrest teaches the organization what not to do next. Every intercepted message forces the creation of a new code. The investigation itself becomes part of the criminal's education. History is filled with examples. The first bank robber leaves fingerprints. The second wears gloves. The third disables the cameras. The fourth never enters the bank at all. Crime evolves, not because criminals become smarter overnight, but because only the adaptable survive long enough to try again. Nature discovered this principle billions of years before humanity gave it a name. Charles Darwin called it evolution. Most people imagine evolution unfolding across millions of years. Dinosaurs becoming birds, ancient mammals becoming whales, slow, patient, almost impossible to observe within a single lifetime. But what if Darwin's theory could unfold not across geological ages, but inside one human body, not over millions of years, but over months or even weeks? Tonight, our investigation reaches perhaps the most astonishing revelation of all. Cancer does not merely survive treatment, it learns from it. By now, our case file has grown remarkably thick. We uncovered hidden identities, forged molecular passports, counterfeit immune commands, corrupted defenders, poisoned environments, energy theft, fortified cities, hidden masterminds. Each chapter revealed a conspiracy of breathtaking sophistication. Yet one mystery continued haunting oncologists around the world. Why do powerful treatments sometimes work brilliantly, only to stop working later? Patients experience dramatic improvement. Scans reveal shrinking tumors, symptoms diminish, hope returns. Then, without warning, growth resumes. The same medicine, the same patient, the same disease. Yet everything has changed. What happened? Imagine federal investigators successfully infiltrating a sophisticated criminal network. Secret headquarters are discovered, communication systems intercepted, dozens of arrests follow. For several months, organized crime nearly disappears. Then, new operations emerge. The criminals now use encrypted applications instead of telephones, anonymous digital currencies instead of cash, different transportation routes, different leadership structures. The investigators did not fail. Their success forced adaptation. Cancer behaves in remarkably similar fashion. Every tumor contains extraordinary diversity. Although cancer may begin from a single transformed cell, its descendants gradually accumulate additional genetic and non-genetic differences. Imagine a city populated by millions of individuals. No two citizens are exactly alike. Some run faster. Some solve puzzles more efficiently. Some tolerate extreme heat. Others survive harsh winters. A tumor contains comparable variation. Most cancer cells share common ancestry, yet each carries subtle differences. Some mutations, different patterns of gene activity, distinct metabolic states, unique survival strategies. Most of these differences prove unimportant until pressure arrives. Darwin described natural selection using a beautifully simple principle. Variation already exists. The environment changes. Individuals possessing advantageous traits survive. Others disappear. Nothing consciously chooses. Nature simply filters. Inside the human body, cancer experiences precisely this process. Treatment becomes the environment. Chemotherapy, targeted therapy, radiation, immunotherapy. Each creates intense selective pressure. Many cancer cells die. Those possessing characteristics allowing survival remain. The next generation arises primarily from those survivors. Evolution has accelerated beyond imagination. Imagine a vast library, millions of books. Most contain ordinary stories. A few describe ingenious escape plans. If investigators burn 99% of the library, the remaining volumes suddenly become extraordinarily important. Cancer follows the same logic. Treatment removes countless vulnerable cells. Rare resistant populations suddenly dominate. Not because treatment created every advantageous trait from nothing, but because it selected those already capable of enduring extraordinary stress. The battlefield itself becomes an evolutionary classroom. For many years, scientists viewed drug resistance primarily as a consequence of new genetic mutations. That explanation remains critically important. Yet modern research paints a richer picture. Cancer adapts through multiple mechanisms. Some cells acquire additional mutations, others alter gene expression without changing DNA sequences. Some enter temporary drug-tolerant states, others rewire metabolism, still others recruit assistance from their surrounding microenvironment. The conspiracy evolves through many pathways simultaneously, like an intelligence organization constantly developing alternative communication channels. Destroy one, 3 more appear. Targeted therapies provide a fascinating example. Unlike traditional chemotherapy, these medicines are designed to interfere with specific molecular abnormalities driving tumor growth. The precision is extraordinary. Imagine investigators identifying the single communication satellite coordinating an international criminal network. Destroy the satellite and operations collapse. Initially, many targeted therapies achieve exactly that. Tumors shrink dramatically. Cancer cells lose essential signaling pathways. Everything appears under control. Then, months later, the network reappears. The criminals have built another satellite, or perhaps several smaller ones. Communication resumes. The organization survives. Researchers began asking a deeper question. Could prolonged therapeutic pressure do more than select resistant cells? Could it actually reshape how surviving cells respond to future threats? Evidence published in Cell Reports Medicine suggests that long-term targeted therapy can foster cellular states associated with heightened stress tolerance and adaptability in surviving cancer populations. rather than remaining passive survivors. These cells may become increasingly prepared for hostile environments. Imagine military recruits enduring months of elite survival training. After repeated exposure to extreme hardship, future challenges become easier. Cancer appears capable of developing comparable resilience, not conscious determination. Evolutionary conditioning. The implications extend beyond one treatment. Some studies suggest that cellular adaptations emerging during prolonged targeted therapy may also influence responses to later immunotherapy. The connection astonished investigators. Imagine detectives discovering that criminals trained to evade financial investigators also became unexpectedly better at avoiding cybercrime units. Different investigators, different methods, yet one adaptation improves survival against both. Cancer's resilience appears remarkably transferable. The lessons learned under one form of pressure may help withstand another. The conspiracy remembers. Consider again Darwin's Galapagos Islands. Different environments favored different beaks, different diets, different survival strategies. Inside tumors, every therapeutic intervention creates a new island, a new ecosystem. Cells capable of surviving that environment flourish, others disappear. The process repeats, again, again, again. Evolution no longer belongs exclusively to ancient history. It unfolds inside living patients, visible through modern sequencing technologies almost generation by generation. Few scientific ideas have transformed oncology more profoundly. Yet, perhaps the greatest misconception surrounding resistance is the belief that cancer becomes invincible. It does not. Resistance often carries costs. A mutation helping one cell survive one therapy may weaken performance under different conditions. A metabolic adaptation improving stress tolerance may introduce new vulnerabilities. Every evolutionary advantage creates potential trade-offs. The detective's task changes. Instead of asking, how do we eliminate every escape route? Researchers increasingly ask, what new weakness has this escape route created? Every adaptation leaves fingerprints. Every evolutionary victory demands payment somewhere else. Modern oncology increasingly embraces this dynamic view. Cancer is no longer regarded as a fixed enemy possessing permanent characteristics. It resembles an evolving ecosystem, flexible, adaptive, responsive. Every treatment changes the battlefield. Every response changes future possibilities. Medicine itself becomes an ongoing dialogue with evolution, not surrender, not defeat, continuous strategic adjustment. Much like intelligence agencies updating encryption after every intercepted transmission. Artificial intelligence now assists researchers in tracking these evolutionary patterns. Single-cell sequencing reveals hidden resistant populations before they dominate. Liquid biopsies detect fragments of tumor DNA circulating through the bloodstream. Mathematical models estimate how evolutionary pressures reshape entire tumor populations over time. The investigation grows increasingly sophisticated. Cancer continues adapting. Science adapts alongside it. The contest has become one of anticipation rather than reaction. History teaches that wars are rarely won by possessing the strongest weapon. Victory belongs to those who understand adaptation. Armies revise tactics. Engineers redesign defenses. Cryptographers replace compromised codes. The side learning fastest often survives longest. Cancer appears to obey the same timeless principle. Its strength lies not merely in uncontrolled growth, but in extraordinary adaptability under relentless pressure. Every failed attack becomes information. Every surviving cell becomes an archive of experience. Evolution writes its report, one generation at a time. As tonight's investigation concludes, Darwin's ideas take on an entirely new meaning. Evolution is no longer confined to fossils buried beneath ancient rock. It unfolds invisibly inside hospitals, inside laboratories, inside patients receiving treatment this very moment. Every therapy becomes both a weapon and an environmental force shaping the next chapter of the conflict. Cancer does not think, it does not plan. Yet natural selection grants it something remarkably similar to learning. The successful survive, the unsuccessful disappear. The result appears almost intelligent, but beneath the mystery lies one of biology's oldest rules: adapt or vanish. Fortunately, evolution never favors only one side. Science evolves as well. Every discovery rewrites tomorrow's treatments. Every resistant tumor reveals another hidden vulnerability. Every apparent setback becomes another clue in the investigation. And in our next and final case file, we will confront the greatest mystery of all: how modern immunotherapy, artificial intelligence, and precision medicine are beginning to reverse the deception, exposing cancer's hidden codes one by one, and turning the body's own immune system back into the master detective it was always meant to be. The conspiracy is vast, but for the first time, its secrets are no longer entirely hidden. Part 11: Turning the Tide. CAR T cells and the rise of biological cyber soldiers. Every great detective story eventually reaches a turning point. After chapters filled with hidden codes, double agents, false identities, and invisible conspiracies, someone finally learns how the enemy thinks. The investigation changes. The detectives stop chasing clues. They begin predicting them. History offers many examples. When encrypted wartime communications were finally broken, entire campaigns shifted. The opposing army did not suddenly become weaker. Its secrets simply stopped being secret. For the first time, its disguises no longer worked. For 10 chapters, we have followed cancer through one astonishing deception after another. It erased identification cards, forged molecular passports, corrupted immune officers, poisoned neighborhoods, starved defenders, stole mitochondria, built concrete fortresses, protected hidden masterminds, evolved under relentless pressure. Each revelation exposed a more sophisticated conspiracy. But every criminal empire eventually faces is a new kind of investigator, one who has studied every trick, one who no longer relies on ordinary methods. Tonight, our investigation enters a new era, not the era of stronger medicines, the era of engineered immunity. Imagine an elite military academy unlike any in history. The recruits are not ordinary soldiers. They are selected from the nation's most experienced special forces. Each has survived impossible missions. Each understands the enemy. But before returning to the battlefield, every soldier undergoes something extraordinary. Their equipment is redesigned, their communications upgraded, their vision enhanced, their mission rewritten. When they return, they no longer search for the enemy using conventional methods. They see through disguises, recognize hidden identities, ignore counterfeit credentials. They have become cyber soldiers. Biology has created something remarkably similar. Its name is CAR T cell therapy. To appreciate its brilliance, we must begin with an ordinary killer T cell. For millions of years, T cells evolved to identify threats by reading molecular identification cards. As we discovered earlier, cancer frequently removes those cards. The detective arrives. The evidence has vanished. The criminal walks away. Scientists began asking a bold question. What if immune cells no longer depended upon those identification cards? What if they possessed an entirely new method of recognition, one that cancer had never encountered during evolution? The answer emerged from decades of molecular engineering. Researchers learned how to remove T cells from a patient's blood, modify them outside the body, then return them as enhanced defenders. Not replacing evolution, extending it. The key innovation lies in something called a chimeric antigen receptor, hence the name, CAR T. Imagine replacing an ordinary security camera with an advanced artificial intelligence system capable of recognizing faces even when criminals wear masks. The disguise still exists. The recognition system changes. Instead of relying solely on traditional immune signaling, CAR T cells receive an engineered receptor specifically designed to recognize chosen targets displayed on cancer cells. The detective no longer waits for counterfeit passports to disappear. The detective recognizes the suspect directly. Cancer's favorite disguise suddenly loses much of its power. The process itself resembles an extraordinary intelligence operation. First, physicians collect T cells from the patient's bloodstream. Think of this as recruiting experienced officers already familiar with the territory. Next, inside highly specialized laboratories, scientists introduce new genetic instructions. The T cells begin manufacturing their engineered receptors. Weeks later, millions of enhanced cells are expanded into an entire biological task force. Finally, they return to the patient. The soldiers have come home. Only now, they possess equipment nature never originally provided. The mission begins again. Early results astonish the medical community. Certain patients with aggressive blood cancers, including forms of leukemia and lymphoma that resisted multiple previous treatments, experienced dramatic responses. Some entered durable remissions that had once seemed nearly unimaginable. These remarkable outcomes led the U.S. Food and Drug Administration, FDA, to authorize multiple CAR T cell therapies for specific blood cancers after rigorous clinical evaluation. The significance extended beyond individual patients. For the first time, medicine had demonstrated that living immune cells themselves could become personalized therapeutic agents. The drug was no longer simply a molecule. The drug was an evolving population of living cells. Yet every remarkable success introduced new challenges. Cancer had spent millions of years learning to evade ordinary immunity. Would it eventually adapt to engineered immunity as well? The answer appears to be sometimes. Some tumors reduce target molecules. Others create suppressive environments. Still others physically exclude engineered T cells just as they excluded natural ones. The conspiracy continues, adapting. But now, the investigators adapt as well. This is no longer a one-sided contest. Researchers across the United States began asking an even more ambitious question. Instead of engineering T cells to recognize predetermined targets, could scientists identify the patient's own most powerful natural tumor-fighting cells, then selectively expand them? Among the institutions advancing this frontier is MD Anderson Cancer Center, where investigators have been developing innovative approaches to identify exceptionally reactive T cells. One example is the ATTACH platform, designed to help isolate T cells showing particularly strong responses against tumor-associated targets for further study and therapeutic development. Imagine searching through millions of detectives, not to recruit new officers, but to discover the handful already capable of solving the most difficult cases. Rather than creating ability from nothing, the system uncovers extraordinary talent already present. The implications are profound. Perhaps every patient's immune system already contains exceptional investigators. The challenge is finding them before the conspiracy overwhelms them. Modern immunology increasingly resembles advanced cyber defense. Artificial intelligence analyzes enormous genetic datasets. Machine learning predicts promising immune targets. Genome editing technologies continue expanding scientific possibilities. Personalized sequencing reveals unique mutations carried by individual tumors. Each patient becomes a separate investigation. Each cancer presents its own encrypted code. The era of one-size-fits-all therapy gradually gives way to precision medicine. Not identical soldiers, precisely trained ones. Yet, perhaps the most inspiring aspect of CAR T therapy lies not in its technology, but in its philosophy. For decades, medicine often approached cancer by attacking the tumor directly. Poison it, cut it away, burn it, destroy it. CAR T introduces another possibility: empower the defender. Rather than building ever larger weapons, build better detectives. Strengthen those already sworn to protect the nation. The strategy feels strangely familiar. History repeatedly demonstrates that lasting security depends less upon stronger walls than upon better intelligence. Of course, no investigation would be complete without acknowledging reality. CAR T therapy is not appropriate for every cancer. Solid tumors remain far more challenging than many blood cancers. Serious side effects can occur, including immune overactivation that requires expert medical management. Researchers continue working tirelessly to improve safety, broaden effectiveness, and extend these approaches to additional diseases. The story remains unfinished. Every breakthrough reveals another mystery. Every solution uncovers another puzzle. Science advances one carefully tested step at a time. Still, the transformation already underway is extraordinary. The immune system is no longer viewed merely as a passive victim of cancer's deception. It has become an active partner in the investigation, programmable, adaptable, trainable, like elite intelligence officers receiving continually updated information. Cancer changes its disguises. Scientists redesign the detectors. The contest becomes one of innovation rather than inevitability. Looking back across our investigation, a remarkable pattern emerges. Cancer succeeded because it repeatedly manipulated communication, false identities, counterfeit signals, corrupted alliances, stolen resources, hidden fortresses, evolutionary adaptation. Every victory depended upon information. Now, modern immunology responds with its own information revolution. Genome sequencing, cellular engineering, computational biology, artificial intelligence, precision targeting. The investigation has entered the digital age, even though its battlefield remains entirely biological. As tonight's chapter draws to a close, one image remains impossible to ignore. Somewhere inside a laboratory, a tiny population of immune cells quietly receives new instructions, not to become something unnatural, but to become something more capable, more perceptive, more resilient. The detectives return to the city they once struggled to protect. The criminals still wear disguises, the streets remain dangerous, the conspiracy continues. Yet the investigators now possess tools their adversary never anticipated. The balance begins to shift. For the first time, the hunters are learning faster than the hunted. And in our final chapter, we will leave today's laboratories behind to confront the greatest question of all. Will the future of cancer medicine belong to engineered cells, artificial intelligence, personalized vaccines, or entirely new biological languages we have not yet discovered? The greatest mystery is no longer how cancer deceives the immune system. It is how humanity may ultimately teach the immune system to see through every deception. The final case file awaits, and with it, perhaps the beginning of the end of cancer's oldest conspiracy. Part 12, A New Dawn. The age of personalized medicine. Every investigation eventually reaches a moment when the question changes. For months, perhaps years, the detectives have asked one thing: who committed the crime? Then suddenly, they begin asking something entirely different. How do we prevent it from ever happening again? The first question belongs to the past. The second belongs to the future. Over the course of our investigation, we have followed one of the most extraordinary conspiracies in biology. We uncovered false identities hidden behind missing MHCI molecules. We exposed counterfeit immune checkpoints. We entered toxic cities ruled by corrupted allies. We watched cancer monopolize nutrients, steal mitochondria, construct biological fortresses, protect hidden masterminds, and evolve under relentless pressure. At every turn, the conspiracy appeared one step ahead. Yet history repeatedly teaches the same lesson. No cipher remains unbroken forever. No secret organization remains invisible forever. Eventually, someone invents a better decoder. Tonight, our final case file does not begin inside a tumor. It begins inside laboratories, data centers, supercomputers, biotechnology startups, academic hospitals, and research institutes where biology and computation are quietly becoming partners. The greatest revolution in cancer medicine may not be another drug. It may be the realization that no two cancers are truly identical. Imagine two detectives investigating what appears to be the same crime. The victims resemble one another. The evidence looks similar. The suspects even share familiar names. Only later do forensic scientists discover that every fingerprint is unique. Every DNA profile differs. Every crime followed a different path. Cancer has undergone a similar transformation. For decades, many tumors were classified primarily by location: lung cancer, breast cancer, colon cancer, brain cancer. Today, scientists increasingly recognize that two patients with cancers arising in the same organ may possess profoundly different molecular landscapes. Their tumors speak different biological languages. Treating them identically no longer makes sense. The investigation has become personal. Perhaps nowhere is this shift more exciting than the development of personalized mRNA cancer vaccines. The word vaccine often brings infectious diseases to mind: smallpox, polio, influenza. Yet cancer vaccines pursue a different objective. Rather than preventing infection, many personalized therapeutic vaccines are designed to help the immune system recognize mutations unique to an individual patient's tumor. Imagine investigators creating a custom wanted poster, not for every criminal, for one specific fugitive. The photograph, the fingerprints, the identifying scars, every detail belongs to one suspect alone. That poster is then distributed throughout the entire police force. The search becomes dramatically more focused. Modern sequencing technologies allow scientists to identify tumor-specific mutations, often called neoantigens, that may serve as personalized immune targets. Messenger RNA becomes the instruction manual. The immune system learns what to look for. The disguise begins to fail. But the future is unlikely to rely upon one strategy alone. History shows that the most successful investigations combine specialists, forensic analysts, cybersecurity experts, field detectives, intelligence officers. Each contributes different strengths. Cancer medicine increasingly follows the same philosophy. Researchers continue developing bispecific monoclonal antibodies. remarkable engineered molecules capable of binding two different targets simultaneously. Imagine a highly trained negotiator standing between two individuals, grasping one hand with each. One side is the cancer cell, the other, the immune cell. The distance separating them suddenly disappears. The encounter becomes almost unavoidable. Instead of hoping immune cells eventually discover their target, the therapy helps bring them together. The meeting is arranged. The confrontation becomes far more likely. Meanwhile, another investigator quietly enters the story. Artificial intelligence, not as a replacement for physicians, nor as an autonomous scientist, but as an extraordinarily powerful analytical partner. Imagine receiving millions of pages of classified intelligence reports. No human detective could read them all. Patterns remain hidden, connections disappear beneath overwhelming complexity. Artificial intelligence excels precisely where information exceeds human capacity. Cancer generates astonishing amounts of biological data: DNA sequences, RNA expression, protein networks, immune cell populations, metabolic pathways, medical imaging, clinical outcomes. Every patient contributes another chapter. AI helps researchers recognize patterns impossible to detect through ordinary observation. Hidden mutations, emerging resistance, potential therapeutic combinations, previously invisible relationships gradually come into focus. The investigation accelerates. Perhaps the most ambitious frontier emerging today is the integration of multi-omics. The name sounds technical. Its philosophy is surprisingly intuitive. Imagine investigating a historical conspiracy using only fingerprints. Important, but incomplete. Now imagine adding financial records, telephone transcripts, satellite imagery, travel documents, eyewitness testimony. Each source reveals another layer. Multi-omics applies the same principle to biology: genomics, transcriptomics, proteomics, metabolomics, epigenomics. Each examines the same cancer from a different perspective. Researchers increasingly integrate these massive datasets into unified models. The result resembles constructing the most comprehensive criminal dossier in medical history. Not one clue, every available clue, together. Yet information alone cannot defeat disease. Action must follow. Researchers are therefore exploring increasingly sophisticated delivery systems. Among the most promising are targeted nanoparticles. Imagine miniature autonomous couriers capable of navigating crowded cities while carrying highly valuable packages directly to a single address. Not every building, one destination. Nanoparticles offer the possibility of delivering therapeutic cargo with increasing precision, potentially improving effectiveness while reducing unintended exposure to healthy tissues. The delivery network itself becomes intelligent. Another remarkable frontier focuses on one of the immune system's oldest alarm systems, the C-gas sting pathway. Earlier in our investigation, we repeatedly encountered cancer's ability to silence alarms. Now researchers ask the opposite question: what if those alarms could be reactivated? The C-gas sting pathway functions as one of the body's innate danger-sensing systems. When appropriately stimulated, it can help initiate immune responses that alert and coordinate broader anti-tumor defenses. Imagine discovering an emergency broadcast network buried beneath an occupied city. For years, its transmitters remain silent. Engineers finally restore electricity. Suddenly, every police station hears the warning simultaneously. The city awakens. Investigators across the world continue exploring how carefully designed sting-activating strategies, often in combination with other therapies, might help transform immune-resistant tumors into ones that become visible and vulnerable once again. Many of these emerging directions appear together in research published across the Nature Portfolio family of journals. One recurring message becomes increasingly clear. The future will almost certainly belong to combinations rather than single miracle treatments. Vaccines, engineered immune cells, checkpoint inhibitors, bispecific antibodies, nanoparticles, innate immune activation, artificial intelligence, multi-omics. Each contributes one piece. The conspiracy became powerful because it evolved multiple layers of defense. Medicine responds with multiple coordinated strategies. This represents something larger than scientific progress. It represents a profound philosophical shift. For generations, a diagnosis of advanced cancer often sounded like the closing chapter of a story. Increasingly, physicians envision something different. Not every cancer will disappear completely. Some undoubtedly remain extraordinarily difficult. Yet many researchers now speak of transforming cancer into a disease that can be monitored, managed, and controlled for long periods, more like a chronic condition than an immediate fatal sentence for many patients, where science continues to expand the available options. That vision is not a promise, it is a direction, one built upon thousands of careful experiments, countless clinical trials, and decades of persistent investigation. Perhaps this is why so much optimism now surrounds the great biotechnology corridors, often compared with new scientific frontiers. In laboratories stretching from academic medical centers to innovative biotechnology hubs, physicians, engineers, molecular biologists, computer scientists, mathematicians, and artificial intelligence researchers increasingly work side by side. The detective story no longer belongs to one profession, it belongs to an entire civilization. Every discipline contributes another decoding key. Every breakthrough illuminates another hidden passage inside cancer's vast conspiracy. As we close the final case file, let us remember what this journey has truly revealed. Cancer is neither a supernatural curse nor an unbeatable enemy. It is an extraordinarily adaptive biological system shaped by evolution. Its strength has always been deception, but history reminds us that deception has limits. Every hidden message can eventually be deciphered. Every disguise eventually becomes recognizable. Every fortress eventually reveals a weakness. Science does not advance because it knows every answer. It advances because it refuses to stop asking better questions, and perhaps that is humanity's greatest advantage. Cancer evolves, but so do we. Not through mutation, through curiosity, through collaboration, through knowledge shared across generations. The investigation continues, and for the first time in history, the detectives possess tools that previous generations could scarcely have imagined. The dawn has not arrived because the mystery is over. The dawn has arrived because we have finally learned how to read the clues. Thank you for joining this journey through one of the greatest scientific detective stories ever told. If this podcast helped you see cancer, the immune system, and modern medicine from a completely new perspective, please take a moment to like this episode if you enjoyed it. If you'd like to explore more long-form science podcasts that uncover the hidden stories behind biology, medicine, evolution, history, and the mysteries of the universe, subscribe to the channel so you won't miss future episodes. And I'd especially love to hear from you in the comments. Which scientific mystery fascinated you the most in this series? What question about the human body, the universe, history, or cutting-edge science would you like us to investigate next? Your ideas often inspire future episodes. Finally, if you think someone else would enjoy unraveling this scientific mystery, please share this podcast with them. Until our next investigation, stay curious, because every great discovery begins with a single question.